Okay. Thank you for that introduction. I am a PhD student from Canada, and it's lovely to be here with you all today. So every day we are bombarded with a chaotic auditory environment full of a multitude of different sounds. But to our brain, not all sounds are equal. For us, vocalizations are one of the most salient sounds. And because of how important vocalizations are for us, our brain has actually dedicated specific regions of the auditory cortex to their processing. Similar to how in the visual cortex, there are specific regions dedicated to facial processing. Now, in humans, these regions are known as the temporal voice areas. There are 3 of them, and they're located along the superior temporal gyrus. These are similar to how, again, in the visual system, there are similar regions for facial processing. Now, as we all know, when we're looking into how a region really works, it can be really useful to have an animal model. For the visual system, macaques were integral to our understanding of how these regions worked at a neuronal level. For these temporal regions, though, the voice patch regions, it becomes more difficult to find a suitable model because this 3-patch system is actually quite unique. Macaques do have one of the voice patches, and this is the anterior one, but they don't have the full 3-patch system. Nevertheless, Pascal Bellin's group in France has looked into how homologous this region is with the human region. To do so, they created this extensive stimulus set that has 4 different categories of stimuli, and it's specifically built for cross-species comparisons of vocalization processing. When they used fMRI to study both the human and macaque anterior voice patch, they found that in both species, the region preferred conspecific noises. However, again, in macaques, studies of these regions are limited to the anterior patch, and to study the full 3-patch system, we need to use a different model. Our group set out to identify whether the common marmoset would be a suitable model for this particular system. We could talk about how cute they are all day, but what's particularly important for them about today's talk is that they have extremely sophisticated vocal behavior. This is an example of what they sound like. It's pretty quiet there, sorry about that. That particular vocalization was from a happy marmoset that was receiving a high-value reward of a pink marshmallow. Don't you dare give them a green marshmallow, though. They will absolutely turn up their nose. A colleague of mine in the lab sought out to identify whether marmosets have a voice patch system similar to the human system. And in fact, they do. So they show 3 voice patches along the superior temporal gyrus, just like us. Now, if it is similar to the human region, we would expect it to show this same trend in the anterior patch that the humans and macaques have shown. And indeed, the marmoset anterior voice patch shows conspecific vocalization preference. And this was an fMRI study done in our lab. So everything so far looks promising. The marmoset voice patches match the human in that there are 3 of them, and they're in around the same regions. But at the neuronal level, do we really know that they are performing the same function? And to answer that question, we need to look a little deeper. Fortunately for me, half this battle has already been won. Zhaojing Wang's group conducted this study using intracranial electrodes in human patients, and they studied the 3 vocal patches in humans. What they found is that the medial patch seems to be an initial hub of vocalization processing, and it responds to vocalizations of all species presented at least. And then the posterior and anterior patches receive information later on, and they respond specifically to conspecific vocalizations. One of my recent goals is to identify whether the marmoset voice patches follow the same dynamics, and I'm going to do so starting with the medial voice patch. To do this, I used NeuroPixel's electrodes, and I inserted them through a small trephination in the marmoset skull into the medial voice patch as defined by MRI coordinates done from our fMRI studies. This is an image of the actual electrode track from which we recorded, taken post-mortem. And the results I'm about to show you today are only from one animal, but we of course have one or two more on the way to replicate the findings. We used the same stimulus set as Pascal's group, and we presented the stimuli in a random order while we recorded from the neurons. And here I have an example of one of the neurons that we found. So along all of the plots, each of the colored lines indicates a different stimulus subcategory that was presented, and the gray line represents sound onset. This particular neuron seems to respond to many of the calls, but has a preference for one of the macaque calls. The second neuron has similar dynamics, but it seems to prefer one of the marmoset calls. And this neuron differs in the way that it responds, in that it has a very quick peak in activity, and then it's actually inhibited relative to baseline. And this neuron is unique from the others, and that it responds significantly only to one of the marmoset calls. So what we can see is that there's a wide variety of responses in this area, and this is what we would expect. However, there's perhaps more non-vocal activity than we would have expected. So let's take a peek at the population data to see if we can get a better idea of what this region actually cares about. One really impactful way of doing so is to do something called population decoding. So when you're doing this, you train a machine learning algorithm on a portion of the entire population of neural activity. Then you give it a small slice of neural activity that it hasn't seen yet, and you ask it to predict what sound the marmoset was listening to at that time. If it's able to accurately predict the stimulus categories, we would say that this region cares about distinguishing that stimulus, and you would see dark cells along the diagonal. So this is about what we would expect for this region. We would expect it to accurately predict all three vocalization categories based on the human data. And in fact, that is exactly what we see. So when we train a machine learning algorithm using the neural data from the medial voice patch, it is able to accurately predict whether the marmoset was listening to human, macaque, and marmoset vocalizations, but it does not accurately predict the non-vocal category. This aligns beautifully with the human data and shows good evidence that this area may be homologous with the human voice area. I'll leave you with that for today, but if you'd like to know more about how these neurons behaved or how the classifier performed on the call-specific data instead of just the species data, I invite you to come to my poster on Thursday. It'll be ID 52.
Yes, there's a question.
The map that you showed for the voice areas, they looked like they spanned like a lot of the auditory cortex. So I guess I'm wondering how specific these voice areas are and what contrast you use to define them. Like do you have acoustic control to control for low-level acoustic features, that kind of thing?
The marmoset ones were defined in fMRI. I didn't do the particular study, but I know that they had marmoset vocalizations, scrambled vocalizations, non-vocal sounds, and I believe other species of vocalizations as well. I don't know, I want to say they didn't have things like pure tones, but I can't say for sure. And we did not use, like in the fMRI figure that I showed, it did have a pretty wide region. What we used for our actual localization was like more central, the kind of brightest activation points.
Great, thanks.
Does that answer your question?
Yes, yes, but let's talk more.
Okay,
yeah.
sure.
Okay, then thank you very much.
Thank you.